yourself in the place of the students. If you're going to come to school 6 or 7 hours a day, and you have the choice between sitting and being a passive recipient of information or actually being a part of it, most kids are going to choose to be part of it.
Two Stripes is the ABE Amgen.
I'm not a book learner, I just can't stand reading. So actually doing the pipettes, actually working with bacteria and different chemicals really helps me learn.
You got it.
We need to get a hands-on experience, otherwise you don't get a good understanding for it. I mean, without actually doing the stuff, there's no point.
The Amgen Bruce Wallace program has been a phenomenal addition to our biology classes here at West.
We're trying to learn about microbiology and also to learn about the techniques used.
It incorporates cutting-edge science for 14 and 15-year-old kids. The students will start with a suspension of what we call competent cells. The experimental tube will have plasmid added to the tube that has had this red fluorescent protein gene inserted into it. And then we're going to try and persuade the bacteria to take up this plasmid.
All the equipment, they're really expensive, so it's the same thing you'd be using in real life.
We do synthetic biology, which is also what researchers are doing in the lab.
The sorts of things that I saw in the Amgen materials were precisely the same sorts of apparatus that we use at Harvard. And exactly the same techniques. It turns out that this is how we do it.
Biotechnology is like super important in medicine, and like this is exactly what we're going to be doing in real life.
So the kind of research we do is a blend of biochemistry and cell biology. We're interested in the way that proteins are produced inside cells because humanity is plagued with protein misfolding diseases, Huntington's, Alzheimer's, Parkinson's. We can't cure these diseases unless we truly understand how these proteins behave inside of cells.
The science that they're doing in the classroom translates directly to the research that's done not only in universities, but in industries as well.
It gives people at that level an understanding of what's involved day to day as a science professional, that the techniques and precision equipment are pretty much the same, whether you're doing them here in industry or in high school. People have the opportunity to try it out and see if they like it.
Keep it slow, keep it steady.
You can get a feel for what the people are doing in the field when you're in 9th grade, you know, so you could figure out early in life, like, oh, is this something I want to do?
We combine the bacteria with the plasmid, and then the kids get petri dishes, and they inoculate the petri dishes with these bacteria and spread the bacteria on the petri dishes.
This is my first time using any biotech equipment, and it's really fun, but it's kind of scary because you have to be so precise.
Can you do it again? Because I think,
Oh, the bubble?
yeah.
Real professional scientists have to collaborate, they have to talk to each other, they advise each other.
So the work environment is very, very collaborative, and we're all very dependent on one another's expertise and input to make sure things go from a discovery stage right the way through to an actual therapeutic.
And you see the same thing in a small setting with the students here in our classrooms. Okay guys, we're going to take a look at the results of Lab 5A. So what do you see on your LB amp plate? Do you see growth on the side that has the plasmid?
Yes.
Yes, what about on the side that doesn't have the plasmid?
Because these labs are based on real science, in other words, they're using real DNA, they're using real cells, things don't always work out the way they're supposed to work out.
What might account for the little bit of growth that's ended up over here? Yeah, maybe. How did you guys distribute the bacteria in these plates when you made them yesterday? You spread them. Is it possibly somebody kind of went that way with the spreader a little bit?
I've seen firsthand what happens when students do make those connections.
A little bit, in fact, a little bit of white growth over here.
You get to learn something that you might not have learned otherwise, and it's very interesting and fun.
How much?
I think it's pretty fun. Hard though.
3, 2, 1.
I love doing science because I think science is an incredible human activity.
All the labs that we've been doing on paper, you can see it finally happening in front of you, and this is cool.
Not that long ago, some of this techniques was Nobel Prize winning.
And it's incredible that all of these things can be done, you know, in a high school setting. That is something we wouldn't have been able to dream about 20 years ago.
Yes.
You guys are done? Fabulous. These can both be welcome white.